Ladies and gentlemen, welcome. For those of you that I have not met, I'm Todd Anderson, and I have the privilege of being the director of the Libin Cardiovascular Institute. The Libin Cardiovascular Institute is a partnership between the University of Calgary and Alberta Health Services, and our mandate is to provide excellence in academic cardiovascular care throughout southern Alberta. And we have about 175 members that are proudly aligned with not only the Cummings School of Medicine, but also many other faculties. The requisite for being in the Institute is an interest and a deep passion for cardiovascular research, education, and clinical care. And every couple of years, we have the absolute privilege of welcoming the best scientists on the planet. And that is absolutely the case this evening, and we're absolutely thrilled that Dr. Eric Olson has joined us from Texas, and we will introduce him in a few minutes. First, what I'd like to do is to introduce Pam Valentine to speak on behalf of Alberta Innovates. Pam is a neuroscientist who we know very well, spent a lot of time at the University of Calgary, and for the last number of years has shepherded us through Alberta Innovates Health Solutions and has been as an important advocate for research, not only cardiovascular research, but all other kinds of research and education in our province. Pam is currently the interim CEO of Alberta Innovates, and she's here to welcome you on behalf of the foundation. Anne.
Thank you very much, and good evening. It's a great pleasure to be, and a privilege for me to be able to be here with you tonight, as I feel that the spirit of this award is an affirmation for what we've worked so hard for to establish here in Alberta, which is that the highest quality of research and innovation should be identified, supported, and honored in our province. Mr. Libin is well known as a longtime supporter of the high-quality research that is carried out at this university and across Alberta, and his generosity has helped us in our cumulative efforts to catalyze research and innovation and the delivery of effective health services and care in heart health. His exemplary philanthropy and volunteerism have helped ensure that we nurture the best health research. As the board chair of the Alberta Heritage Foundation for Medical Research from 1990 to 2000, Mr. Libin led the foundation through some truly significant years in our history. It is because of the strength and capacity of our health research community that we are building toward an even more robust era of health research and innovation in this province. To mark Mr. Libin's legacy and recognition of his contribution, AHFMR supported the creation of the Alvin Libin AHFMR Award in the Cardiovascular Sciences at the University of Calgary. The intent of the award was to honor a leading thinker and innovator in the area of cardiovascular research and healthcare. Generous contributions by Mr. Libin's friends were also instrumental in developing this award into what it is today. Alberta's support for research, along with other investors, was key to building a provincial community of top cardiovascular researchers, clinical researchers at the University of Calgary and at the University of Alberta. This community has in turn attracted funding support from key people like Al Libin, who know and support quality when they see it. The HFMR endowment attracted research talent to Alberta and continues to fund top quality health research and innovation initiatives through Alberta Innovates. At Alberta Innovates, we support a broad range of activities, from discovery to application. Alberta Innovates is about building on our province's strengths in the health sector, as well as environment, energy, agriculture, forestry. We are working with our partners to diversify Alberta's economy, to improve our environmental performance, and to enhance our well-being. We are here to be the catalyst to deliver 21st century solutions to the most compelling challenges that we face as Albertans today. On behalf of our board of directors and the staff at Alberta Innovates, I would like to offer my sincere congratulations to the 2016 recipient of the Alvin Libin AHFMR Award in Cardiovascular Sciences, Dr. Eric Olson. Thank you.
Pam, thank you very much. It's now my real pleasure to introduce Dr. John Meddings. John is the Dean of the Cummings School of Medicine. He's just passed his 5-year mark, 5 down, 10 to go. John, for those of you who don't know, is a wonderful leader, has really spearheaded our movement through the naming of the medical school, accreditation at the medical school, and really responsible for the theme as we move forward in precision medicine, and he's here to bring greetings from the Cummings School of Medicine. John, thank you.
So thank you, Todd, and I won't be long. I'm here to welcome everybody, both on behalf of the University of Calgary, but particularly the Cummings School of Medicine. And I thought I'd just tell you a little bit about the Cummings School of Medicine. One of the things that we talk about, and you'll hear us all the time talking about, is creating the future of health. It's what we wish to do. And as Todd has mentioned, is one of the obvious questions from that is, what is the future of health? And I'm here to tell you that the future of health is something that we call precision health or precision medicine. We're going to hear some things about that today. And in some ways, I think the easiest way of describing precision health and precision medicine, why is it different than what we do today, is that precision medicine means that one size does not fit all. That there are individual differences. There are individual differences in our biology, in our genome, in our epigenome, in other parts of us that affect our own individual disease. And that in order to really treat somebody, you need to take into account those individual variations when you make a diagnosis, when you prescribe a treatment, and perhaps most importantly, when you actually want to prevent illness. So that's what we're doing. And in order to get there, what we're doing is investing in what we think are the most important things. We call them the 3 Ps. They're people, they're platforms, they're partnerships. Most important are people, and we're investing in some of the best scientists, some of the best clinicians in the world, bringing them to Calgary. We're also investing in platforms to make those people aspire to work as hard as they possibly can. And perhaps the most important part are partnerships. They're partnerships with Calgarians in the room. They're partnerships across the province of Alberta. They're partnerships around the world. In fact, a little later this week, we're presenting to the government of Alberta a strategy for precision health or precision medicine actually for the province. And it's critically important that we involve the province. Our ability to be successful at that is going to be through our institutes. And I would be remiss if I didn't say how proud and pleased I am with the Libin Cardiovascular Institute, with Todd Anderson for his incredible leadership of this institute. It's been an amazing journey. And I thought I would just say a couple of words about Alvin Libin. And Pam, you preempted most of what I wanted to say, so I'll be fairly brief. But I wanted to say thank you, Alvin. I wanted to publicly acknowledge some of the things that you've done. I think people often don't know this, but we have unbelievable health care in Calgary today. In fact, if you look at cardiovascular care, I would hope that nobody in this room is going to have a heart attack. But if you do, Calgary is probably the best place in Canada, if not North America, to have one. And you might ask, why is that? And the why of that goes all the way back to what Pam described as the Alberta Foundation for Medical Heritage Foundation for Medical Research, AHFMR. AHFMR was a creation of Peter Lougheed many years ago as a premier of Alberta. And one of the things that it has done over the years is recruit to this province some of the world's best scientists, best clinicians, and because of that, we have some of the world's best healthcare here. That was a tough job. And I want to acknowledge the fact that Alvin Libin chaired the board of the Alberta Heritage Foundation through a very difficult time in the 1990s. So we owe you a lot of thanks, actually, Alvin. This institute, which bears your name, has done some unbelievable work in the past, over the past decade. It publishes about 350 papers a year. It's brought in about 150 million in research over the last 10 years. It's done some phenomenal stuff, thanks to some amazing people here in the leadership of Todd. So I'd like to thank you for that, Alvin. It's been truly amazing. And Dr. Olson, I'm looking forward to this. It's wonderful. Having spent 4 years of my life in Dallas, I know the temperature differential you've come through to speak to us at. I really am looking forward to your talk. Thank you for coming to Calgary.
John, thank you very much for that. Approximately 1 year ago is that the Libin Executive Committee set out to pick the 6th HFMR Libin Prize winner. And we'd had 5 distinguished clinician scientists, really leaders on the planet, starting with Dr. Eugene Brunwald. These individuals write textbooks, they produce large numbers of papers, and really set the stage for clinical cardiovascular care. When we assembled what we thought was a list of the best scientists on the planet, is that the unanimous winner of this award was Dr. Eric Olson. The committee came together, they looked at his attributes, people had heard him speak, and they recognized the great changes that had occurred in our knowledge based on his work over the last 20 or so years. So Dr. Olson is a basic scientist, molecular biologist, currently is the chairman of molecular biology for the last 20 years. So for you department heads in the room, is that he's now into his 5th term as the department head, and shows no signs of slowing down. And he's been at UT Southwestern since 1995 or so. And really what he's done in his research laboratory with the number of students who've gone on to spectacular careers is really defined how heart and other muscle cells develop. Embryological development has been important, what the genetic regulation determinants are of muscle cells, not only of heart, but also muscle cells, as you're going to hear tonight, that cause diseases of the muscle. And he's adopted therapeutics into those regimes. He started 3 companies as a result of his translational research. We measure greatness in academics based on how much money you bring in and what you produce to tell the world of your research. Well, Dr. Olson is a giant in that field, produced over 700 publications. A spectacular career is when your publications have been cited 10,000 or more times. His have been cited some 90,000 times. He sent me a copy of his CV, and I thought, well, this isn't that long. I mean, I've seen what he's produced, and there was 6 pages of publications. This was 6 pages of publications in Nature and Science, okay? So this is like the cream de la cream. Most people, if they had one of those in their career, would be absolutely ecstatic. He's had more than 40 in the last 25 years. Every 1 or 2 years, his lab produces a scientific manuscript that will change the way that we think about muscle disease and cardiovascular disease. His list of awards going forward is really superlative. Not only does he win awards as a distinguished scientist, most recently he's won an award from the American Heart Association as a distinguished mentor. So he treats his students very well. I asked him about his lab today. He said his lab contains about 40 people, and on any given day he would know what all of them are doing in his lab, and that includes Saturday and Sunday. So he's been very successful because of that great combination of innovative thought, incredible hard work, and diligence to what he's doing. And so we couldn't be more proud that Dr. Olson accepted our invitation to come to Calgary to be the 6th recipient of the Libin HFMR Prize, and we're delighted to hear from him. He's got a provocative title on heartbreaking and ways to heal the heart. Dr. Olson, welcome to Calgary.
Thank you, Todd, for that gracious introduction. I'm really happy to be here, and I first want to thank the Institute for choosing our work as deserving of this award. I'm truly honored to receive the Libin Prize, and I want to congratulate Mr. Libin and all of you on the extraordinary environment that you've created here. I just had a terrific time today talking to people. There's really great science, and there's a palpable enthusiasm in this institute, so congratulations to all of you. I should also say I felt kind of like, with just the attitude and just the environment, I felt sort of like I was in the Texas of Canada up here. Alberta sort of feels like that, or maybe Texas is the Alberta of the United States. So I'm really happy to be here, and my lab works on a lot of things, as you heard from Todd, and I hope that you're not disappointed, but at the last moment I changed the topic of my talk tonight, so I'm not going to talk about heartbreaking and heart-making, which was the title of the talk, but I'm going to talk about something that I thought might fit with the interests that John expressed about precision medicine. So this is an area that I'm very interested in, and so I'm going to tell you about that tonight. As you heard, my group works on the biology of muscle. We've worked on this for 30 years or more. The students in my lab discovered many of the key gene regulatory proteins and molecules and mechanisms that form all the muscles of our bodies. To put this work into context, muscle is the meaning of life. There is no activity by any animal since the beginning of time that occurred without muscle. I mean, from the ceaseless beating of the hummingbird wings, which beat 85 times per second, to the movement of the largest animals on Earth, to the beating of the heart every second from 2 weeks after conception until the end of life, to all movement of the body, all art, all music, all athleticism, everything originates from muscle. My neuroscientist friends don't like it when I say this, but I'll say it anyway. There is no output of the nervous system without muscle. In fact, you can function without a brain. Teenagers do it all the time. But there is no life or meaning to life without muscle. So there are three major muscle types in our bodies: skeletal muscle, cardiac muscle, and smooth muscle, schematized here. And as I said, my students and postdoctoral fellows have used a variety of genetic and biochemical approaches to try to understand what are the genes and what are the mechanisms that orchestrate the formation of these three muscle types during development. And they've used a variety of model organisms, ranging from fruit flies to mice, amphibians, and even studied humans. And along the way, they uncovered many gene regulatory networks that evolved to form these different muscle cell types. And these are ancient, very ancient networks that date back more than 600 million years ago to the appearance of the first muscle cells in evolution. And they have been maintained because of the importance of muscle in all of life. But many of these developmental pathways and mechanisms we now understand are redeployed in adulthood and serve as the platform for many disorders. And so that by understanding the fundamentals of developmental biology and tissue formation, one can not only understand the molecular basis of disease, but one can exploit those molecules and mechanisms to regenerate tissues and organs. So I'm going to focus tonight primarily on one muscle, but I'm also going to talk about the heart. But the muscle I'll talk about primarily tonight is skeletal muscle. This is the largest muscle in all of our bodies. Counts for about 40% of the body mass. It's also this major source of whole body metabolism. And it's also the most regenerative of all tissues. We all hear about regenerative medicine. Well, muscle epitomizes regeneration. And you can see that here. This is a histological section. This is what normal muscle looks like. This is muscle from a mouse, but human muscle would look much the same. It's highly organized for contraction. But in response to injury, muscle will rapidly degenerate. You can see that here in this panel. This is muscle from a mouse that's been injected with a snake toxin, and it causes the fibers to essentially disintegrate, and then there's a lot of infiltration. But you can see just 3 weeks after this destruction of muscle, the entire muscle is completely restored and reconstituted and is fully functional. It's only distinguishable from the original uninjured muscle by the presence of a central nucleus in each new muscle fiber. And we know a lot, which I won't go into tonight, but we know a lot about the cell biology and the molecular biology that underlies muscle regeneration, and this is due to stem cells that reside in adult muscle. But muscle is also the source of many of mankind's most insidious diseases, and I think it's remarkable to realize that there are more than 750 monogenic disorders that lead to the loss of function of muscle, both skeletal muscle and heart. So these are disorders that are caused by single mutations in the DNA code that ultimately are manifested by the destruction of muscle tissues. And there are more than 400 different genes that are known to be mutated that lead to neuromuscular disorders. There's not a single cure for any of these disorders, and these are the worst disorders of mankind. So given the long experience in my laboratory of studying the biology of muscle and the mechanisms of muscle growth, development, and regeneration, we, in the past few years, have decided to turn our attention to try to correct some of the most insidious of all muscle disorders, and perhaps the worst of all is a disorder called Duchenne muscular dystrophy, or abbreviated DMD, that many of you may have heard of. This is a disorder, as we'll talk about, that's passed on from mothers who are carriers of a mutation which they're unaware of until they have a son who's affected. So these carriers, the female carriers, will typically have a healthy boy until about the age of 2 or 3, and then typically the mother notices that the boy is faltering on the playground or perhaps having trouble walking up the stairs or standing up, and typically the mother will take the child to the pediatrician, who will ask the son to perform what's called a Gower's maneuver, in which they'll ask the boy to stand up off the floor, such as this, and inevitably, because of weakness in the limbs of these boys, they use their hands to stand upright. And subsequent to this, the disease is ultimately diagnosed. So this disorder affects 1 in 5,000 boys. There's 300,000 boys in the world right now suffering from this disorder, and there is absolutely no cure. Once the disease is diagnosed, there is no escaping the inevitable consequences of it. And this is the progression of the disorder. Typically, as I said, these boys are first diagnosed, noticed by their mothers at age 2 or 3. They have walking problems, and this soon progresses to a complete loss of ambulation due to disintegration of the muscles of the legs. They become wheelchair-bound. Then they lose use of their arms. They ultimately are on ventilators due to an inability to breathe. And the ultimate cause of death, which occurs in the mid-20s, is heart failure. So cardiac muscle cells ultimately fail in these boys, and that's the cause of death. One of the other horrific aspects of this disorder, which I learn about in a poignant way because I literally receive letters every single day from mothers on all corners of this earth who have boys that are recently diagnosed with this disorder. And because this disease is often not diagnosed until these boys are sometimes 3 or maybe even 4 years old, it's not uncommon for a family to have had a second son prior to the diagnosis of the first. And then they, of course, face a quandary. Do we get our second son genetically tested? And when do we tell him about the situation with the disease? Because if he has the mutation, he will ultimately develop these symptoms. So we know the cause of this disease. It's caused by the largest gene in the human genome called the dystrophin gene. And the dystrophin gene is encoded on the X chromosome, which we'll talk a bit about. This gene is the largest gene in nature. It's a massive gene. For the molecular biologists, I know we have a mixed audience here tonight, but for the molecular biologists in the audience, I'll just tell you, it takes 18 hours to make 1 copy of this gene into an RNA. So this is an incredible feat of molecular biology. This gene encodes 2.6 million bases. It's absolutely massive. And it contains, as we'll talk about, 79 exons, which I'll tell you what those are for the lay people in the audience, that encode 3,500 amino acids in a massive protein. So we've known about this gene. Lou Kunkel at Harvard discovered this gene about 30 years ago. We've known that this is the gene which, when mutated, causes this disorder, but there has been no headway towards finding a cure for this disorder. This shows the inheritance of this disease. So if we have a mother and a father, the mother here is the carrier, and I've highlighted the mutation in the dystrophin gene in red. So, as I think most of you know, females have 2 X chromosomes, and males have 1 X chromosome and a Y chromosome. So when this couple has offspring, and the mother passes an X chromosome down to the sons, 50% of her boys will have an X chromosome from her that has the mutation, and 50% will get her normal X chromosome. And 50% of the girls will have an X chromosome with the mutation. They'll be carriers, and this girl will not carry the mutation. But because females have 2 X chromosomes, even though she's got the mutation, she has the other X chromosome, which has a normal dystrophin gene, so she doesn't manifest. Same with her offspring, but since boys have only 1 X, every boy that inherits this mutation will develop the disease. There is no escaping that diagnosis. It's become apparent in recent years, and we have a clinic now in Dallas, that it was previously thought that female carriers were asymptomatic, but it's now clear that by age 50, female carriers develop cardiomyopathy and heart failure because of the absence of 1 copy of dystrophin in their hearts. So the heart is very sensitive to this protein. So this is a muscle tissue, what it looks like. And the dystrophin protein, without going into too many details, is schematized here. Essentially what this protein is it's a molecular shock absorber for muscle, and it sits right below the surface of the muscle membrane, and it connects an adhesion complex at the cell membrane with an underlying scaffolding. And you can think about your muscles, all the muscles of your body and your heart are contracting constantly, and the dystrophin protein holds the membrane intact. And when the dystrophin protein is missing, after the persistent contraction of the heart and muscles, the muscles begin to leak, and then they ultimately disintegrate and collapse. And this is what the dystrophin protein looks like. This is a cross-section through muscle tissue, and the dystrophin protein, we've stained it here in green, and you can see that just beneath the surface of every muscle fiber is located this protein, where it serves as the stabilizing force. So to drill down a little more deeply on the structure of the dystrophin protein, think about it as a shock absorber here, and here's the big protein, and it has the 2 ends are essential. So these are like the 2 ends of the shock absorber. They anchor this protein to the different parts of the muscle membrane, and the central part is the shock absorber with the springs, and it's kind of redundant in this region. You only need a few of these springs for the protein to work. And we know that because there's a large population of patients in the world that have a mild form of muscular dystrophy. It's called Becker's muscular dystrophy, or BMD, and they have the 2 ends of the shock absorber, and they got a few springs in the middle, but they don't have all of them. And so you can see that here. And these patients, they have a very mild form of muscular dystrophy. These patients can live into their 70s, they're ambulatory, and it's really relatively a minor disorder. So it's been thought that if one could convert DMD into BMD, that it would be a clinical triumph, and it would be life-changing for these boys and for these families, and that is what we're trying to do, and that's what I'm going to tell you about for the rest of this lecture tonight. So over the past 30 years, there have been heroic efforts to try to find any sort of cure or effective therapy for these boys that are affected with this disorder, and all of them have failed, and this schematizes some of the therapies that have been tried. One has been to try to introduce dystrophin by gene therapy back into these patients by putting it into a virus and asking, or other vehicles and asking, can you put the gene back? It can't work because the gene is so big. It's the biggest gene in nature, and you can't transfer a piece of DNA of that magnitude back in. There's been other approaches to skip mutations. There's been approaches to express compensatory genes. There's been a lot of highly publicized efforts to transplant normal muscle cells or stem cells into patients with Duchenne muscular dystrophy. I'm here to tell you this is never going to work because 40% of your body is muscle, and you can't transplant enough muscle cells to reconstitute that. And one thing we've learned from stem cell clinical trials over the past decade is that cells don't colonize the heart, and so you can't restore cardiac function by transplanting stem cells in this disorder. And then there have been other drugs that have been tested. Some of you may have read recently, there's been a lot of publicity on this in the United States recently, about a clinical trial for a drug called Eteplirsen, which was developed by a biotechnology company called Sarepta Therapeutics. And what Eteplirsen is is a short oligonucleotide sequence which masks an exon, a segment of the dystrophin gene, and skips over the mutation to put the gene, basically put the gene back together. And in principle, this works well, but in practice, there was no evidence for efficacy of this drug in boys that were treated with this drug. And the way this drug is delivered is these boys get a central line permanently placed into their chest, and then they go in weekly and have this oligonucleotide delivered directly into their chests. It was an open trial, no controls, with less than a dozen patients, and no evidence for restoration of dystrophin protein expression in these boys. But the FDA approved the drug anyway because there was nothing else that could be done for these boys, and now this drug is being provided to boys with a specific mutation in the gene in exon 50, which we'll talk about. The drug costs roughly $400,000 per year per patient in the United States. And that is the only available therapy for these families or these patients. And so there are many other approaches underway, and there are some promising advances, and hopefully those will move forward quickly. We have decided to take a new approach and ask, can we permanently correct the mutation responsible for this disorder? And to do that, we want to use a very new technology. The scientists in the room are familiar with it. The rest of you may be less familiar with it. It's called gene editing. And actually, when I got on the plane last night, the plane was delayed, so I went to the magazine shop and picked up a magazine. Here's the first magazine I saw there. So Gene Genie is in Newsweek. And so this is an article that talks about this technology. It's called gene editing. And for those of you who haven't heard about this, it's the most revolutionary technology that I've seen in my career, and I believe it's going to change the future of precision medicine and genetic disorders in general. So how does this work? Let's talk about the human genome, which I've schematized here. So the human genome, every cell in our body contains 3 billion base pairs, letters of DNA. That is a lot of letters. How many letters is 3 billion letters? You want to know how many letters that is? 3 billion letters is the same number of letters in 1,000 Bibles. So that's a whole lot of letters. And what this new technology of gene editing can do is it can seek out a single letter that's wrong, and it can correct it with absolute precision and fidelity. So you just think about one misspelled word in 1,000 Bibles, and it can find it immediately, and it'll change it back to the right spelling. That's pretty good. So here's how it works. Suppose you have a mutation in a gene, one letter misspelled in the dystrophin gene, since that's what we're going to talk about. So what this can do is it can find that letter, and it can correct it. And how does it do this? This is totally amazing, actually. So this is a bacterial immunity system. So bacteria developed this technology to eliminate viruses that infect bacteria, and they can use it to correct their genome. And so how does it do it? It works by, you can provide a single RNA called a guide RNA, and this has complementarity to whatever sequence in the genome you want to target it to. And when the guide RNA goes to the sequence that you want it to go to, it will bring along with it an enzyme called Cas9, which is an enzyme that can cut the DNA at that particular site. I showed that here in scissors. And when Cas9 cuts the DNA at that specific site, the mutation can be either cut out or it can be corrected. And the DNA can be put back to whatever sequence you want it to be. So this is really incredible technology with respect to its efficiency and simplicity. So just for the scientists in the audience, I need to say a few more technical terms. So this is schematically how it works. Here's the enzyme. It's called Cas9. It goes and it cuts the DNA at any sequence where you want it to cut if you provide a guide RNA. And then There's one other sequence that's required for cutting. It's called a PAM sequence. It's just this sequence. So it's AG or GG adjacent to the guide. It'll cut at that site. And you've got to remember that the AG sequence is really important for what I'll talk about later. And so then if you provide with this enzyme and this guide RNA a piece of DNA, it'll insert the DNA at that site and it'll correct it perfectly through a method called homology-directed repair, or HDR. So you can put any sequence into DNA with absolute precision, or you can correct a mutation perfectly. Or if you don't provide the DNA template, you just provide the enzyme and the guide RNA, it'll cut the DNA, double-strand DNA, it'll just chop it. It's not precise how it cuts it, so it fills it back in. The machinery in the cell fills it back in and leaves what's called insertions or deletions, but just tiny little modifications of the DNA. So we decided to ask, is it even feasible that one could use this revolutionary technology to correct a mutation responsible for Duchenne muscular dystrophy? And so to do that, we started with a mouse that has the disease. It's called the MDX mouse. It's the most commonly used model for this disorder. It has one mutation, a single letter in the 23rd exon. Exons for the non-scientists are exons are the segments of DNA that make an RNA that's spliced together to form the ultimate protein. So we asked if we could correct that. And to do this, we used a technology called germline editing. You'll be reading about a lot of this type of editing in this big article in that article in Newsweek. They talked about this yesterday. So what you do in this is you take fertilized eggs like you would do in a in vitro fertilization clinic, and you inject those eggs with the gene editing components, Cas9, the guide RNA, and here we use the wild-type oligo sequence for that site, and we inject them into the eggs, and then you put the eggs back into a mother, and then she gives rise to offspring. And when these offspring are born, their genes are corrected. And so we looked at these mice when they're born by looking at their muscles, and here you can see the muscles that are stained for dystrophin. So in a normal mouse muscle, all the fibers are green. They're all expressing dystrophin, and in the MDX mouse, they have no dystrophin at all, so that's why they have muscular dystrophy. And then we found out that when we did this approach, you generate what are called mosaic animals. These are mice that have different degrees of correction in these early animals, because it's a little bit stochastic how it occurs early on. But that really turned out to us to be an unexpected bonus, because we could then ask, what is the minimum level of editing of this mutation that would be necessary to cure this disease, at least in this animal model? And so this was an animal right here. This animal had only 17% correction. That's a pretty low level of correction, but it had almost complete restoration of dystrophin-positive muscle fibers. Not perfect, but pretty good. And then this animal had only 41% correction. It's perfect. It's completely rescued. It's been estimated that in boys with Duchenne, if you could correct about 15% of the mutation, that you could cure the disease. So it's not going to be necessary to bring dystrophin all the way back up to normal. Even a relatively modest correction may be sufficient to provide the cure. So these initial proof of concept studies were very encouraging, because they at least said, yes, it's possible in an animal model through germline editing to correct the mutation, and you don't even have to be very good at it. Even a little correction gets you there. And we've done a lot of subsequent editing. You can also, the way boys are typically diagnosed is through a blood test. In the pediatrician, they take a small sample of blood and they measure an enzyme in the blood called creatine kinase. This is a muscle-specific enzyme, and if you have Duchenne, your muscles are leaking. And so creatine kinase is pouring out into the bloodstream, and that's how they diagnose it. So you can do that the same in mice. So here's a normal mouse that has no creatine kinase in its bloodstream. Here's the MDX mouse. It has roaring levels of creatine kinase in the bloodstream. And then here's 3 mice that we corrected by gene editing. This one had 17% correction, 41% correction, 83% correction. And you can see it pretty much brings these animals right back down to normal. So that's really encouraging. So one important take-home message from what I've told you so far is that the efficiency of correction of the gene at the mutation, that the correction of the disease is greater than the efficiency of the correction of the mutation. So even if you correct just a little bit of the mutation, you have a really dramatic effect on the correction of the disease. And we wondered why that is, and I think the answer lies in the unique biology of muscle. This is what muscle cells look like in a dish. And all the muscles of our body have hundreds of nuclei in a common cytoplasm. They're a multinucleated tissue. And so if you could correct the gene for dystrophin mutation and even a few nuclei in a common cytoplasm and they make them make dystrophin, then the dystrophin can kind of diffuse through the whole fiber and correct it. So we think that makes muscle really susceptible to this gene editing. So when we published this first paper, there were a lot of excitement, but a lot of issues were raised about this, appropriately so. And these are the 4 major issues, and for the rest of the talk, I'm going to tell you how we're trying to confront these issues. The first one's a big one. Germline editing is not feasible in humans. Well, it's feasible, but it's not ethical, and it's not going to be happening anytime soon, at least around here. So germline editing means correction of sperm and eggs, like in the whole germline and all the cells of the body, like we did in those first proof of concept studies. So you got to get past that. You got to have a way to correct the disease in the person that has the disease, not in the genes that person's going to pass on to humanity. Homology-directed repair that we used in the original approach does not work in post-mitotic in tissues that don't divide, like heart and muscle. That's a huge one. Thousands of DMD mutations exist in the world. So this comes back to your precision medicine issue. There's literally 3,000 different mutations. So how can you correct all those? And then how are you going to, if you can't do this in eggs, and you have to do it in boys, how are you going to deliver all this gene editing machinery into all the muscle cells of the boy? So here's, again, the dystrophin protein schematized, and here's the structure of the gene. So this gene has 79 exons. For the non-aficionados, again, the exons are the segments of the DNA which, when their products are spliced together, give rise to the final protein. And you can see the different domains of the protein color-coded onto the exons that encode them. And here, the shapes of these exons indicate whether their splicing together maintains the production of the protein across that region. And so as long as the shapes are compatible, that means you have appropriate splicing between them. So we looked at this, and this is the MDX mouse again. It has a mutation in exon 23 that we'd already corrected. But now we wondered, could we do it simpler? What if we just make 2 cuts in each of the flanking sequences, the intronic sequences, and just chop out the exon and let these 2 splice back together? And that would not require the homology-directed repair. That could be just an imprecise cut in each intron. So we did that. Here, dystrophin's in red, normal, MDX, no dystrophin, and here's 2 mice that we corrected. So this is really easy now. You can just skip an exon that's got a mutation and put the gene back together. And here's dystrophin. This animal has only 10% correction by skipping, but it's pretty good for dystrophin. And this one has half the genes are corrected, but it's completely back to normal. So then we looked at this, and now I'm just going to say a few words to the molecular biologists in the audience, because I think this is really amazing. We looked more carefully at the PAM sequence. Remember, this is the recognition sequence that Cas9 identifies in DNA, and it'll cut when there's the appropriate PAM, and the sequence was AG. It turns out AG is the universal splice acceptor sequence for splicing of all exons in the genome. So every exon is preceded by an AG. So that meant you could direct Cas9 to any exon that you want to skip. You just get a guide RNA here, and it'll cut at the AG, and then you can delete the exon. To me, this is amazing. This is like one of the most amazing inventions of nature, that nature created a bacterial enzyme for immunity in a bacteria, and that enzyme is perfectly adapted for recognizing splice junctions in mammalian exons for exon skipping. So nature really is inspiring sometimes. So, okay, then how do you deliver all this to an animal, a mouse, or eventually a boy? So for this, we asked if we could engineer a harmless virus called adeno-associated virus, a virus involved, I guess, could cause the common cold. We just took the harmless portions of this AAV, and we put into the virus the gene editing components, Cas9 and guide RNAs, and we asked, what if we just inject, and we use a specific form of AAV called AAV9, which infects only muscle cells and heart in the body. It's a really interesting virus. It'll just go right to muscle. So we said, what if we inject mice with this? We tried a bunch of delivery methods, asked, could this work to correct the disease? And AAV is really interesting because it's been approved by the FDA for use as a delivery vehicle in people because it's harmless. So here's the result. Here's some normal mice. They have dystrophin, of course, in every fiber. Here's the MDX. They have no dystrophin. Here's an animal that we injected just into the hind limb muscles with the virus. And we looked 3 weeks and 6 weeks later, and lo and behold, we could see the appearance of dystrophin-expressing muscle fibers. Even 25% of them were expressing dystrophin. So then we thought, well, why don't we just go the whole body? So we did intravenous, one delivery, intravenous, and we could see the appearance of dystrophin-positive fibers. It's not perfect, at least it wasn't then. It's getting there now. But here you could see the appearance of dystrophin-positive fibers. This is really, to me, amazing, that you can take an animal, an adult animal, that's never made this essential protein for its muscle, and with a single cut in the DNA of this animal, you can now convert it to an animal that makes the missing protein and restore that protein. And you can measure the strength of a mouse. Mice aren't very strong, but you can measure how much force they can exert with a machine. So a normal mouse, let's say, exerts 70 grams of force, and the muscular dystrophy mouse has about 35 grams of force. And here, when we did gene editing, this was 4 weeks after systemic delivery, we could already see the appearance, the strengthening of the muscles due to the production of dystrophin, and this gets better over time. Okay, so that looks pretty good, but MDX mice have only one mutation, and it's not even a common mutation that you see in people. People have more than 3,000 different mutations. So here's how those mutations are categorized. So a third of them roughly are point mutations, that one single nucleotide change that disrupts the protein. These are the 79 different exons that are spliced together here. And 2/3 of these patients have deletions of exons that put the protein out of frame, and then a small number have duplications that are very interesting from a molecular biology point of view. So again, here's the exons, 79 exons, and this is what's called a hotspot, exon 50. That's the #1 hotspot mutation site in boys with muscular dystrophy. The most common site of mutation in the world is exon 50. And so what happens is, in these boys, this short segment of the gene is deleted, and that means 49 exon splices to exon 51, and you can see the shapes are incompatible. So it basically terminates the protein in the middle, and so this essential end of the shock absorber can't be made, so the protein's dysfunctional. This is the exon that the Sarepta eteplirsen drug is directed against trying to fix. So we wanted to ask, can we fix this human mutation, the most common human mutation? Of course, there wasn't a mouse that had that mutation, so we made a humanized mouse. So we again can use gene editing, and in this humanized mouse, we just deleted exon 50 from its genome, and then again, 49 goes to 51. It's not compatible. So this mouse, here's what it looks like. Here's a normal mouse, and here's this delta exon 50 mouse. It has no dystrophin at all, and you know that creatine kinase enzyme in the circulation now is diagnostic for muscular dystrophy. So this delta exon 50 mouse is roaring for creatine kinase in its bloodstream. So this mouse is a really interesting platform for therapeutic testing of any types of therapies for correcting this humanized mutation. So we asked, could we correct it? We refer to this method now as myo-editing or muscle editing. Could we correct it by skipping exon 51? You can see when you skip exon 51, then 49 can splice to, can't read that from here, 52, puts the protein back in frame because those two exons are compatible. And yes, it worked. Amazing, it worked. So here's dystrophin, normal. Here's the humanized mouse, has no dystrophin. And here we did one single injection. My students were really getting good at this method by this point. So here they did one single intravenous injection. I hope you can see that 4 weeks after injection, all these muscle fibers, not every one, but pretty good, all these muscle fibers were corrected and making dystrophin protein. So we also want to look at the heart, because the heart's what ultimately kills these boys, and you can see that here. So here's normal heart, all the heart muscle cells are dystrophin positive. The humanized delta 50 mouse has no dystrophin positive, and here's an animal 4 weeks after injection systemically. The majority of the cardiomyocytes are now expressing dystrophin. And so we can quantify that. This is a gel. So here's the two animals, dystrophin proteins, very large protein. Here's the humanized mouse, no dystrophin protein, single injection, 4 weeks of the virus with the gene editing components, 4 weeks later in the heart, dystrophin protein, brought back almost to normal levels. So again, we think 15% of normal level would be curative for this disorder. This is bringing it back to about that, well, way beyond that level, actually. So I think this is encouraging, but there's still a ways to go. So this, again, is the structure of the gene, 79 exons I've schematized here, and there are hot spots where the gene is more susceptible to mutations than other regions, and those hot spots cluster in these two regions I've shown here in yellow. So about 80% of the mutations in the world of boys with DMD are in these two regions of the gene. And this shows the top 10 hotspot exons and of all the mutations. And so we focused on the top 4, which account for 35% of the mutations in the world for DMD are in these 4 exons, and we've made humanized mice now for all of those mutations. So we can take those humanized mice and we can test our exon skipping strategies for correction in those animals. These are the top 12, top 12 exons, hotspots highlighted, and those are the ones that account for about 80% of the human mutations. And we've developed now optimized guide RNAs that can skip each of those 12 exons. So if a patient appears, we know his mutation. If it's in one of these 12 exons, we have the optimized guide RNA that can deliver Cas9 to skip that exon and put the protein back in frame. Okay, so now the last part of the talk, I want to tell you about our initial attempts to try to move this towards humans. So we have a muscular dystrophy clinic in Dallas, and so here's the way it works. Patient comes in, and we can take a small sample of his blood, and we can determine the mutation in that boy. Or I should say that, I mean, for all the many, many, many letters I get from around the world, even in the outer recesses of India or places where you would think, you know, they may not have the best medical attention, almost all mothers know the mutation of their boy. So that in just about every case, they'll email me, so my son has this mutation, can you correct it? And I'll say, I'll tell them if we can or can't. In some cases, if it's a really interesting mutation, we will go get a blood sample and bring the blood back. And from the blood of the patient, we can create what's called iPS cells, induced pluripotent stem cells, so you can use a technology to make permanent stem cells from the blood, and then you can convert them into muscle cells in a dish, cardiac muscle or skeletal muscle in a dish. And then you can test these optimized guide RNAs against the genome of this patient. And you can ask, can you skip the exon and create dystrophin protein? And sometimes if we get a letter from someone and, you know, we can't get their blood or whatever, now, in fact, you don't even need their blood. You can create the mutation of the boy, and you can just start with normal iPS cells, and we can use CRISPR-Cas to make the mutation that we want in the boy, in the cells from normal, so they're just like the boy, and then you can try to correct them to restore dystrophin. So here's how it works. So this is a real boy, And this boy, here's the hotspot region I told you about around exon 50. So this boy has a deletion. This boy's from Japan, actually. He has a deletion exon 48, 49, 50. So his exon 47 splices to 51. Again, this is the big hotspot I've been talking about. So 51's incompatible with this, so when it splices there, it puts the protein out of frame. And so we made iPS cells and then cardiomyocytes from this boy, and they're shown here, and they're stained in red, and there's no red, so they can't make dystrophin. So we asked if we just used a guide RNA to deliver Cas right against this exon or splice junction here and destroy it, and then let 47 splice to the next one, and you see the shapes are compatible. So here's what happens. Here's the cardiomyocytes from this boy stained for dystrophin. So, I mean, this is really remarkable. One single cut, it doesn't even have to be precise. You just cut and erase the AG sequence of the splice acceptor of that exon 51, and then let the splicing machinery take over, and it splices these two exons together, and now you get intense red staining for dystrophin. So this boy's cells, this boy's body, and this boy's cells have never made dystrophin protein. And with this single precision medicine genetic step, now they make dystrophin protein. And so here's a protein blot. Here's dystrophin protein. Here's uncorrected cells from this boy. They make no dystrophin. If you cut here and skip the exon, now they make dystrophin protein. And they make it at the normal level, too, because you're correcting the mutation of the gene in its normal environment. So all the regulatory mechanisms that drive the expression of dystrophin are now driving the expression of the corrected dystrophin. So it expresses at the same level. And so cardiomyocytes in boys with Duchenne or in mother carriers when they reach their 50s, they become arrhythmic. I'm not an electrophysiology guy, obviously, but even I can tell that's not too normal. So these are 4 cardiomyocytes individually patch clamped. If we're looking at calcium flux, you can see what it looks like there. And these are from this boy, that they're arrhythmic, and then we did the single step of gene editing, and they're converted to rhythmicity because they make dystrophin protein, and it basically seals the membrane back up, and it stabilizes the structure of the muscle fiber. Okay, so this is a source of one source of my inspiration. This is my neighbor. His name is Ben. He has muscular dystrophy. He's in a wheelchair, and it's his mother, Debbie, and his cardiologist. So Ben, he's in his early 20s. We looked at his dystrophin gene, and we know his mutation. He has a single nucleotide change in an intron of his dystrophin gene, and it's just one letter. Think back about this 1,000 Bible analogy. He has only one little change. It's a T that's changed to a G, and it's not even in the protein coding region. It's in an intron sequence, which, a lot of people say it's garbage sequence. It's just in between these two coding exons. But that one nucleotide put him in a wheelchair because AG is the universal splice acceptor sequence for all exons. And so that T to G mutation created a false splice acceptor sequence. So 47 splices to a pseudo exon, a fake exon, 47A that has a stop codon. And so when that splice occurs, he can't make the other end of the dystrophin protein. And so he has muscular dystrophy. So we looked at this, and this was really remarkable to realize that inside his disease lies his cure, because his mutation, T to G, created an AG. It created a disease-specific recognition site for Cas9. So Cas9 can be directed specifically against his mutation in that intronic sequence by using a guide RNA adjacent to the AG. So we believed that we could erase that G just by Cas9 editing and put the protein back in frame. That's exactly what happened. So here's dystrophin protein on a gel in a normal person. Here's Ben, he has no dystrophin. And then here we corrected it by erasing the pseudoexon, and now you have dystrophin protein. And here we stained it in green. We made cardiomyocytes from him and stained them for dystrophin in green. You can see this one's making dystrophin. And we had Ben come up to the lab, look through the microscope. So not very many people in the world ever get to see their own beating heart cells in a dish. And I can assure you not very many boys with DMD can see their beating heart cells making dystrophin in a dish that their body never made. So it was very, very exciting to see that. So these then are the types of mutations that exist in these patients, and these are the potential, this shows the potential to correct these mutations. So we believe that this gene editing strategy can correct nonsense mutations, mutations that impose a premature termination on the protein, like in the MDX mouse. It can do that by skipping the mutation and putting the protein back in frame. And when you do that, it's not a perfect correction. You restore the two ends of the protein, but the middle is a little deleted. So it's essentially like we talked about, it's converting DMD into Becker's or BMD. If there's deletion of various regions, and this is like the humanized exon 50, which puts the protein out of frame, you can delete an ensuing exon and put it back in frame. I showed you evidence of that in that one Japanese boy, partial rescue. You can correct duplications where certain exons are duplicated and it changes the reading frame. I didn't talk about that today. Those are really interesting because when you correct this, by removing the duplicated exon, you create a perfect scarless dystrophin protein. And then there are pseudoexon mutations like Ben has, which impose a false exon in the middle that stops the protein. And when you correct that by skipping, you put the protein right back together, perfect, seamless. So that's really amazing precision medicine, at least in principle, to recreate the normal gene following a single genomic editing step. So I'm always asked, are there mutations that can't be fixed by this approach? We believe between 60 and 80% of the mutations in the world can be fixed in principle. It hasn't been shown yet, but in principle by this approach. But there are some mutations that cannot be fixed. If one has a large genomic deletion that eliminates either end of the shock absorber and the DNA is not there to edit any longer, if it's just deleted, you can't fix it. So you've got to have something to work on to fix it. And that's a small percentage of patients, but a significant number. And those patients will likely be eventually treated by various gene therapy approaches, such as the delivery of truncated versions of dystrophins or other proteins that several labs are doing great work on. So finally, what are the, I think it's worthwhile to talk about, what are the distinguishing features of gene editing compared to other therapies that can be brought to bear on patients with monogenic disorders. Again, this is in principle. In principle, this approach leads or can allow for permanent correction of disease-causing mutations because it eradicates the cause of the disease. The disease is caused by a genetic mutation. If you get rid of the genetic mutation, there's no longer a disease driver. And we believe in principle that one-time treatment could be sufficient. And I think for disorders like of muscle, in particular of heart, this will be the case. Because one thing that's been clear in recent years from work from a number of laboratories is that the heart cells that you're born with are the heart cells that you die with. Heart cells last your whole lifetime. And so if you can, muscle cells, skeletal muscle cells probably last most of your lifetime too, although there's not been we don't know that as certainly as we do for the heart. But so if you can correct a mutation in a cardiac muscle cell nucleus early in life, it's likely that that correction will withstand the test of time. This approach can potentially reach all the affected cells if it's efficient enough. Certainly in mice, it's efficient enough. And using the approaches I've described, there is the potential to correct up to 80% of DMD mutations. That's still got to be proven. Certainly it'll work in a dish, and it'll work in the humanized mice that we've generated so far, but we haven't obviously generated mice for every type of human mutation. So this is, I think, my final slide. This is our vision for applying gene editing technology to Duchenne muscular dystrophy. I've told you that we can certainly correct mutations in boys, in cells from boys in a dish as proof of concept to show that this truly does work. We can humanize mice so that they harbor the same mutation as patients, and we can correct the mutations in those humanized mice by delivery of gene editing components in a virus that has specificity for infecting muscle cells. So the next challenges are to scale up. Can we do this in larger animals or not? We'll see, as a prelude to moving towards humans. And we also need to carry out safety studies, probably in non-human primates. So lastly, what are the challenges and potential concerns that scientists and clinicians need to be mindful of in advancing this type of technology? Now, keep in mind that this technology was only first described a few years ago by Jennifer Doudna, Emmanuel Charpentier, and others. So this is the pace at which this technology has moved is really breathtaking, and I think we still don't, it's not even out, we're still at gene editing version 2.0. I mean, we don't know how far this can go, but clearly it has room for optimization. So one of the common concerns that's frequently mentioned is the possibility of off-target effects of gene editing. So that is to say, what if when you're targeting a specific mutation, the machinery inadvertently targets another gene? What if that gene is a gene that's important for life? We really have to be cautious about that. We and others have looked carefully for off-target effects in animals. We have not observed them. No one has yet reported any pathologic off-target effect in an animal that I'm aware of. We certainly have not seen any ourselves. The method is very specific, but in cultured cells in a dish, people have seen off-target effects with high doses of gene editing components. So this is really an important point. But I think ultimately, with a disease like Duchenne or other diseases like this for which you know the consequence of the diagnosis, it may ultimately be important to balance the risk against the certainty of the progression of the disease, and those are decisions not to be made by me, of course. So another issue to consider is immune responses. Cas9 is a bacterial protein. Will it evoke an immune response, or even dystrophin is a foreign protein in Duchenne boys? Will that evoke an immune response? The dystrophin immunity is an issue that will relate to all forms of monogenic disorders for gene therapy. And I think both of these, I'm optimistic, hopeful, let's say, that these can be mitigated by immune suppression. And all these boys anyway are on low-level immunosuppression to begin with. The longevity of rescue, we've talked about that. Can a single correction last a lifetime? Even if it can't last a lifetime, can it extend or delay the onset of the disorder? I think if the earlier the intervention, the more likely it is that one can extend the quality of life and delay the inevitable with this disorder. And then there are other challenges these are more just practical challenges of scale up and delivery. So I want to close by talking about the people in my group that did this work. So our work on gene editing was done by a graduate student, Chengzu Long, in my lab. Chengzu, I try to encourage my students to take on big, risky projects, and Chengzu was a real bold guy, but he worked in my lab for 3.5 years, and he couldn't get anything to work at all. I mean, he was trying to do big things. It wasn't his fault. And so one day he read this, when the first paper on gene editing came out, where they were just talking about gene editing to inactivate as a strategy that could be used to inactivate genes. It was a paper published in Cultured Cells. He came to me and he said, why don't we turn this around and see if we could not inactivate genes, maybe we could use it to correct genes. So he said, what disease could we apply that to? And I thought, DMD is the obvious one because we know all these mutations. So he really boldly took that project on, and he did a great job on really developing that. He has his own lab now at NYU. Leonella Amawasi is a postdoc in my group who did all the viral work on restoration of dystrophin. And these other students and postdocs have participated in the humanized studies and the work on iPS cells. We've had many collaborators in Dallas and elsewhere that have enabled these studies. I have a long-term colleague, Rhonda Basil-Dubey, who makes things work in my laboratory. We have an exciting muscular dystrophy clinical research center at UT Southwestern, shown here. And here's my laboratory in Dallas. And with that, I'm happy to take any questions, and thank you all for your attention.
I think we have about 5 minutes for questions before we do the presentation.
Excellent job. I really enjoyed this. I have just 2 questions. You briefly touched upon the exon 6, 7, 8 mutations. That falls under the active binding mutation. So, do you think skipping the exons there would really affect its binding capability? And
So let me answer the first question first,
Okay.
and then I'll do the second, so I'll forget the first one. So you're asking a really good question. The actin binding domain is a very sensitive, fragile domain of dystrophin. And I think we don't totally know how the piecing together of different segments of that might or might not reconstitute the dystrophin protein. So I have a postdoc in my group, Victoria, and she is focused exclusively on the actin binding domain region. So she has obtained a large collection of human samples of patients with different deletions in the actin binding domain, and she's also made humanized mice with the actin binding domain deletions. We can correct several types of mutations in the actin binding region, but right now we're looking carefully at the stability of the proteins that come out from those corrections and other things. But there will be some actin binding domain deletions that cannot be correctable, but I'm optimistic that several of them can be corrected.
And this is really a selfish question. I'm just starting my lab at the Libin Institute. So can this technique be really utilized on smaller proteins? This is a protein with 79 exons. Let's say I'm working with a protein with 11, 12 exons. So skipping exon there would really not be a best approach, right? Or just your thoughts on that?
I think it's a good question. It depends on the protein. In order to do exon skipping, you have to be able to eliminate that one segment of the protein and retain function of the rescued protein. So I think if there are exons that can be eliminated and still restore function, it'll work. If there's exons that can't be, then another approach would have to be taken.
Other questions? Maybe I'll ask one myself. So Eric, spectacular lecture. In your best estimation, when do you see applying these to human studies?
That's always the question I don't want to try to answer, but I will say that we have to do the large animal proof of concept, and that's underway right now. I think that will be done in 2017. And we need to do a safety study in primates, which I think can be done. I think if everything works, if we can scale up, if we can maintain efficiency, and paramount, if we can confirm absolute safety of the technique, I think within a few years we could envision doing some sort of simple trial to see if it could work. It's very important that everyone proceed with caution with this technology, not just us but others, because this is at the outer edge of technology. I mean, it's making changes in the human genome, and it has enormous potential, but it requires enormous responsibility, because if there's a reckless mistake, it could set this technology back. So I think it's important to be careful. But I'm sure you can appreciate the pressure brought to bear from families to move this technology forward, especially when they see their sons deteriorating and they know there's nothing for them. So, yeah, I'm very sensitive to that, and we're working as hard as we can to try to move this forward.
A few questions for us. You mentioned that AAV9 virus is specifically for skeletal muscle and cardiac muscle. My question is that whether your virus can infect this smooth muscle, or are they cells?
It's highly selective for, it's the most selective for heart. The heart is really sensitive to infection with AAV, so that's why the editing in the heart's so effective. Skeletal muscle is also very effective. There are reports that it can infect smooth muscle. We looked in our first paper at whether we saw correction in vascular smooth muscle cells, and we reported that we did see it, but we haven't looked at that in great detail yet, so I don't know what the efficiency of that is, but it can work there. But as you know, there's a whole spectrum of adeno-associated viruses with different tropisms, so there may be others that might be more amenable to vascular editing.
Dr. Olson, I just wondered if you encountered any religious problems with your research. Have you come across any problems of that nature?
You mean religious resistance to gene editing, or? I've not faced any, but I think it's there are certainly ethical issues. I was at the National Academy of Sciences, and there was a lot of discussion about the ethics of germline editing, and I don't know if that touches on what you're talking about, the religious aspects, but certainly my personal point of view is that it's too early to think about germline editing, and it's not up to us to make decisions about who gets germline edited or not, because once the editing occurs in the germline, those changes are permanent for the flow of genes through humanity for all time. And so I think right now, all responsible scientists are thinking about this in terms of somatic cell editing of diseased tissues. But certainly you can imagine that the technology works you could do germline editing right now, but I think no one is appropriately known as doing that as yet.
Final question.
So that was a great talk. Thank you for coming and sharing that with us. At the beginning of your presentation, before you got into the really exciting cutting-edge work, you did allude to the recent FDA approval of the one drug. And I wondered now, in the context of hearing everything you said, if you could just tell me what I missed the first time in terms of how that worked in contrast to what you're doing now. And in particular, you mentioned there was a lack of dystrophin, and it wasn't clear to me what the drug did that allowed it to get approved. It must have done something good for someone, or so the FDA thought.
So the Sarepta drug is based on skipping of exon 51, the exact exon that I showed you that we are working on skipping by gene editing. And the way that it's a short oligonucleotide sequence that masks the splice acceptor sequence of 51 and allows skipping then to the next one to restore dystrophin. I was not involved in any of that, and I'm not an expert. I only know what I've read in the lay press and in various journal articles. The FDA asked that evidence for restoration of dystrophin protein be provided as a criteria initially for approval, and they were unable to obtain evidence for elevation of dystrophin protein above 1% with a standard error equal to that, so they couldn't show that. There were reports that a subset of the patients may have had delayed progression of disease, or I believe a subset had some improvement of 6-minute walk test, but there was not, to my knowledge, statistically significant evidence for improvement, and in particular, the key point is, can the drug do what it's supposed to do, which is to restore dystrophin, and that was not able to be demonstrated. Now, maybe it does restore dystrophin and they weren't able to show it for some reason, but nevertheless, the drug was approved based on that small number of patients. And whether that sets a new precedent for drug approval in the United States for other indications or for dystrophin, I mean, I told you today there's 12 exons that account for 80%, up to 80% of the patients. And if you can develop oligos for addressing all those, the market for those, of course, is huge. And the cost is huge too. So I think there's many approaches. There are obviously many approaches. In particular, I think the microdystrophin approach that's being tried by several groups, that is using the same AAV viruses that I described to deliver the truncated version of dystrophin to the cells of the body. There's clinical trials ongoing with that approach, and I think that looks really promising. It's just a different approach than the one we're taking, and perhaps someday there'll be a combination of approaches that will be used.
So an absolutely spectacular talk, even for me who understands nothing about molecular biology. I now understand gene editing, the best talk I've ever heard on that. I would like to close the evening by asking Alvin Libin to come forward and make the presentation. Alvin, as you are aware, has done so many wonderful things, and really the link with the community, with Alvin's family, has made the Libin Institute run, has made the Libin Institute thrive, and for that we are eternally grateful. So it's always fitting that we can do this. We're going to do this every year or two, and wonderful that we can be having Dr. Olson come forward to it. So I'd like to ask Alvin to go forward and say a couple of things and do the presentation to Dr. Olson.
Well, Eric, thank you. That is incredible stuff. You know, for a layperson, it's really the opportunity and what's in the future is something special. So thank you for coming to Calgary. Thank you for coming to Alberta. I understand you're going on to Edmonton. And thank you to Pam for your nice words, and Dr. Meddings and Todd. It was an excellent evening. We have a couple little gifts here for you, and here's one of them. And we've got a book that we wanted to give you that is the cardiovascular history of what's going on in Alberta.
Thank you very much.
So thank you very much.